Okay, so proteins are made up of amino acids. This is the basic shape of what the amino acid looks like at pH, physiological pH 7. You've got the acid end is deprotonated, and that proton goes over to the amino end and forms an ammonium ion. Amino acids, there are 20 of them, and they can be broken up into 4 groups, and they are basically nonpolar. That means they're only groups that are alkanes. There's neutral. When I say neutral, that means no charge. Neutral and polar, for example, we have alcohols or thiols, and then there are basic and acidic ones. And again, these side groups totally determine the shape of a protein. And that shape, as you know from, we talked about Monday, we talked about the ammonium ion in an acidic solution will end up being very positive. When we change that acidity to on the alkaline side, that charge goes away. So then the shape of the protein would then change. And because many enzymes, excuse me, back up, all enzymes are proteins, that means enzymes are also sensitive to those same pH and temperature changes. And on the enzyme, now we're going to have a whole unit on enzymes. So basically, every enzyme has got something called an active site, and that's where all the magic happens. That's where the reactions are catalyzed. So if the shape or conformation of that active site changes shape, then the enzyme doesn't function as well or not at all, depending on how drastic the active site changes. Okay. Now this is glycine. This is the one that's got is neither P or L, and all the rest of these are L. These are L, 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 so on. Okay, so these are all neutral. Basically, we've got just a methyl group here, an isopropyl group here, an isobutyl group here, and just all nothing with any charges. Neutral and polar. Okay, typically these are involved with some kind of acid or alcohols. And nitrogens can do hydrogen bonding, and that's why they're considered polar. And of course, we have a thiol here, which acts similarly to an alcohol because in the periodic table, the S is underneath the O. Now, for your next test, I'm going to let you have a half page of notes again, just like the test 2. And these are all basic side chains. And of course, because they're very ionic, they're very polar. And the same goes for the acidic ones. Remember, ionic is the most polar you can get. Okay. So again, the ethyl alpha carbon is chiral. So we can have a D or L form. So the natural one is the L form. And glycine is not chiral or achiral, meaning it doesn't have 4 independent groups attached to that carbon here. Now this is, again, is the alpha carbon. And that's where the side chains hang off of. Now, as I said in the beginning, at physiological pH, this acid here is in its ionic form or deprotonated form. Well, that protein ends up migrating over to the amino end. And because it's a proton with a positive charge, that nitrogen becomes an ammonium functional group with a positive charge. So does anyone know German? I believe Zwitter means something like both versions existing. So a Zwitter ion will be both a negative and a positive charge on it. So this is the way we draw amino acids in here, like that. Now there's something called, which is really an important characteristic of both amino acids and proteins, something called the isoelectric point. Okay. Iso means the same. So basically, at the isoelectric point of an amino acid or protein, this applies to proteins as well. Like there's a nice lab I have where you determine the isoelectric point of an amino acid that we're not going to do in here because of the other school. Anyway, basically when the pH is at the isoelectric point of the amino acid or protein, that means everything cancels out. So the net charge then is 0. Now, if you think about a protein like milk, okay, protein in milk is casein, and its isoelectric point is around 4. That means that pH values, let's look at casein here. So if we have a pH greater than 4, that protein is a net negative charge. We have a pH of less than 4. That charge is positive. And then a pH of 4, net charge is 0. So when you have a glass of milk, okay, what do you think the pH of your glass of milk is, approximately? Okay, to answer that question, is your milk sour, neutral, or bitter?
Neutral.
Most of the time it's neutral, unless it's been sitting out too long. Okay, so if your milk is at room temperature, or not room temperature, take it out of the refrigerator, it hasn't been sitting around for hours or days, Do you see any clumping in your milk?
If it's been sitting around for a while, yes.
No, just pour it out of the carton right into your glass. Is it clumping
From the fridge, or
from the fridge? Yeah,
it shouldn't be clumping, no.
right. Okay. So in other words, the casein protein is dissolved in the water. Okay. Now your pH is greater than 4. It's around 7. Because if it tasted sour, its pH is less than 7. If it tasted bitter, the pH is greater than 7. So we look at taste now. When you taste something, you can tell whether the pH is less than 7, greater than 7. Let me give you an example. If you take a corn tortilla right out of the package and eat it, it tastes bitter. The reason why it tastes bitter is because the corn in the masa flour has been treated with slaked lime, which is a base or an alkaline substance. So your corn tortilla are basically above pH 7 because they're alkaline, hence they are bitter tasting. When you cook them, you neutralize them, and basically you come up with more of a neutral, so that bitterness goes away. Vinegar. Vinegar, as you know, is acetic acid, hence the pH is going to be lower than 7. Hence, vinegars taste sour. Okay, so let's go back to our milk now here. You take your milk out of the fridge and it tastes fine. There are no clumps. That's because no casein lumps or casein precipitates. Why? Because the protein is negatively charged. Therefore, soluble in water. Now, if we leave it out for a couple weeks, that glass of milk, it starts to clump up. Why do you think that is? Has anyone made cottage cheese? No one in here has made cottage cheese? Well, if you haven't made cottage cheese before, you should try it. Very easy to make. This is the way you make cottage cheese. Usually whole milk. Okay, what happens when we add lemon juice to the pH?
It decreases.
Exactly. So the pH goes down. And guess what? When it reaches pH 4, That's the isoelectric point for casing. So the charge on the casing now at pH 4 is neutral, 0. Everything is cancels out. So that protein is no longer negatively charged. Hence, it's nonpolar. So what do nonpolar things in water do?
They do not dissolve in water really well.
Exactly. So it clumps. And that's what cottage cheese is. It's when we bring the pH down to 4 by adding lemon juice. What happens is we reach the isoelectric point of the casein protein, and there's a number of proteins in there, but in general, they're around 4. It becomes insoluble in water and clumps up. Then you pour off the water, which is called whey, and what we have here is called curd. That's how you make cottage cheese. Now let's go back to our glass of milk. Does that make sense, you guys? Okay. So let's look at our okay, what's going on in that 2-week-old glass of milk after a couple, 3 weeks?
It smells nasty.
Mm-hmm. Yeah. What else? What does it look like?
It's clumpy.
Lumpy. Okay. So you have a bacteria called assuming I spelled bacillus right. So okay. Lactobacillus bacteria. When they eat things, they produce lactic acid. That's where the name lacto comes in. So we have these lactobacillus bacteria sitting there chowing down on that old milk. The more they chow down, the lower the pH goes until it gets to 4. The protein is no longer negative. Its net charge of 0 and becomes insoluble in water. So it clumps up. That's why old milk got clumps in it, because the pH has been lowered down to the isoelectric point of the casein protein. Now you make cheese. Lactobacillus bacteria makes cheese. So now you've had a little food science with the protein. All right, does that make sense? That isoelectric point. Now, in biochemistry laboratories, one way to separate proteins is by modifying the pH so that one of them approaches the isoelectric point of that one protein and it becomes insoluble and precipitates out. Then you filter it. Now you've separated one protein from another based upon pH and their isoelectric points. It's also a way to determine the isoelectric point for the pH for that protein, is to keep changing it until it precipitates out, then you know the isoelectric point for that protein. Okay, so isoelectric point's a big deal. So make sure you understand that concept. Okay, okay, so basically Zwitter ions are ionic, and they tend to act like ionic solids, high melting points, soluble in water, and they form a white crystalline solid. But the main point is, because it's ionic things have a much higher melting point, and they're also very soluble in water. Okay, we have one amino acid, cysteine, which has a thiol group in it. If we oxidize, and remember, when we oxidize organic compounds, we remove hydrogens. Which is the same as losing an electron. Okay, so the hydrogens we lose are these two on the thiol groups. So this goes from cysteine to cysteine. This is a covalent bond. If this was in a protein, this would be a covalent crosslink. It's the strongest crosslink there is a covalent crosslink. The more covalent crosslinks you have, the more rugged the protein is to things like change in solvent and heat. Now, this relates to hair. The protein you have in your hair is made up of disulfide bonds. Now, these determine curl of your hair. So when you get a perm, what has to happen is you have to break the disulfide bonds. So step number one for a perm, and we want to change the location of the disulfide crosslinks in a perm. So step number one is we want to take reduce the disulfide bond by adding H's or adding electrons. So it becomes now two separate amino acids rather than one. Now your hair is going to go limp because there are no crosslinks that define the curl. And what you do now is you're going to set your curl. That means you're going to make it curlier or straighter, but whatever. Then we're going to oxidize we're going to reoxidize the sulfide bonds to the disulfide bonds, creating a permanent curl or straightness, whatever you change it to. That's why when you get a perm, for those who have had a perm in your life, they're really stinky because reduction and oxidation reactions tend to have lots of foul odors. And the reason why it's a perm is because we have disulfide cross-links, which are covalent, are very, very strong. The only way you can get rid of a perm without going through this reduction process is letting it grow out. And so you have the new hair, which has the genetically based disulfide bonds in it and a genetically based curl. So this cysteine amino acid is a really important amino acid. Now your book will show this looking like that, which is not true. This is what it looks like. This is an L and this is an L. This is an L and this is a D. And whenever there is a amino acid in your body, it's going to be the L form, not the D form. So this is for publishing purposes, the one on the left. Okay, so before I get into that, I'm going to talk about this a little later, but basically, here. The cysteine thiol group bonds very strongly to heavy metals. So things like mercury, things like lead, etc. Those bond permanently. So if that cysteine amino acid is part of an enzyme, then that enzyme is going to be disabled because that thiol group can no longer function as a thiol. Be bound up with the mercury or the lead. This is why heavy metal poisoning is so bad for you, because it binds up enzymes. So the enzymes are no longer working. If the enzymes are doing important work in your body, then that doesn't happen anymore. Okay, so that's about talking about the individual amino acids. Now, as you remember from Monday, to make an amide, we need some kind of an acid and an amine. We end up with an amide. Now, in protein lingo, they call that a peptide. But it's in OChem lingo, it's an amide. Same thing though. So all the reactions that work on an amide also work on a peptide. And this is the basis by which proteins are synthesized using the whole, DNA, the whole transcription process going through the DNA, where you have the mRNA and the tRNA, and you have all these enzymes, RNA units that are coded to specific amino acids as the protein is being built. But the bottom line is that enzymatic process creates this amide bond. Now, whenever we make a reaction of amino acids in a test tube, not enzymatically, but in a test tube, you have 50-50 chance of it hooking up like this. Like we were mixing glycine and alanine together. Okay. The odds are 50-50, you're going to get one of each. Either the glycines first or the glycine is second, just 50-50. Okay, that would be on the bench. Or in situ. Okay, if we do it enzymatically, we get 100% of one or the other. Okay, it's real important to distinguish between these two. Enzymes are horrendously specific. Very, very specific. And it will not be a 50-50 mixture. If an enzyme is involved, then it'll be one or the other. I can't emphasize that enough. Enzymes are horrendously specific. It all happens in the active site. That active site is modified, then that specificity goes away. Okay, now when we start adding amino acids together, so if we have one amino acid, it's called amino acid. If we have 250 amino acids, it's called a polypeptide. If we have greater than, let's say, 50 to 51, it's called a protein. Okay. Now there's a convention that everyone adheres to when we put the ammonium ion on the left and the carboxylic residue on the right. And it's just the way it's been doing. So that includes writing an individual amino acid or a polypeptide or a protein. N's go on the left, O's go on the right. So proteins now, greater than 51 amino acids, take the form of Zwitter ions. That means the beginning is the amino group at the beginning is and got a positive charge and the end got a negative charge. Because that hydrogen migrated to there. If we're looking at glycine, for example, say we had 1,000 glycines altogether. It's going to look like this. So this is the alpha carbon. And then we have, let's put the nitrogen here. It's going to be bound up to get more of there. Let's say we had, again, 1,000 glycines altogether in a nice long, real simple protein. Okay, at the beginning of the protein, on the N terminal end of the protein, that's going to look like And the very end, that's the alpha carbon. So. That makes up the Zitter ion. The one end, it's got a positive charge and 3 ammonium. Other end is a carboxylic acid salt, negatively charged. Proteins, like I was mentioning before, amino acids and proteins both have an isoelectric point. They can behave as buffers. If you remember from 305, a buffer will take a hydroxyl ion and neutralize it, or a hydrogen ion and neutralize it, so the pH doesn't change. And there's a range where that works. Can be in solution or form stable colloidal dispersions. Okay. What that means is it clumps. Okay. The form of that clump is going to depend on it's going to depend on the side chains of the amino acids in the protein. So you can have, just a big, it's 20 amino acids. So you can have, acidic one, basic one, polar neutral, or neutral nonpolar. And that ratio is going to determine the isoelectric point. So when the net charge is 0 at the isoelectric point, proteins clump and precipitate in solution because they're now no net charge, hence they don't dissolve very well in water. Okay, there are 2 main types of proteins: fibrous and globular. Fibrous are long, when you think of hair, you think of muscle fibers, meat, those are fibrous proteins. Globular proteins are the opposite. Fibrous proteins tend to be very rugged. Globular proteins tend to be very delicate. What I mean by delicate, that means so they're sensitive to heat and pH changes. Now, when you cook a piece of meat, it changes form. The fibrous proteins are rugged, but not indestructible. So heat and pH, like when you do, you can, what is it? The Mexican dish. Oh, I can't remember it now. When you cook shrimp in acid citrus.
Ceviche, Professor.
What is it?
Ceviche.
Ceviche, right. So to make ceviche, you put some kind of protein in there, and you're adding citrus, usually lemon, orange, whatever. And what that does, that lowers the pH significantly, and that protein then denatures, changes shape. And it quote unquote cooks it versus changing the shape via heat. So who likes ceviche? I really like it. I frequently will order it at a restaurant. Okay, let's talk about globular proteins now. Globular proteins are usually instead of long and skinny, they're typically globular, kind of roundish, big kind of glob, and it has to do with two things. Solvents. What solvent is the globular protein in? Is it polar or non-polar? Secondly, the amino acid side chains. So what will happen is if the amino acid side chains are in general polar, and your solvent is polar, usually water, then the amino acids will, the polar amino acids will tend to migrate towards the outside. And the polar, non-polar, so this, they will migrate to solvents. The non-polar side chains will migrate away from solvents. So if we have a polar solvent, usually water, for the amino acids with ionic, the acidic, basic, and the neutral polar side chains will all migrate outward towards the water, and the non-polar, neutral non-polar side chains will migrate inward, and that will create the shape. It's solvent and side chains together. Solvent is very important. Change the solvent, the shape of that protein will change. Like if you went to a non-polar solvent, this protein over here would be turned inside out because the non-polar inner part would migrate to the outer part close to the non-polar solvent. That's why changing solvents will denature a protein and stop an enzyme from working because the active site is changed. The shape of the active site is changed. If you change the pH of the solvent, that interaction changes again. If you make the solvent basic, then the acidic parts of the side chains will be attracted more to the solvent. If you make it acidic, then the basic part of the side chains will be attracted. If you made it the solvent alkaline, so the opposites attract. Okay, so proteins can get very, very, very complex. When you're in graduate school, and you're interested in being a biochemist, you are typically very, and when you're working with proteins, you're very, very specific about that. Because there's such a variety of proteins, man. Also, proteins can act together with other proteins. For example, this here we have 4 proteins that are acting together as one unit. You see the different colors is a protein. They can sometimes act in conjunction with other molecules, like hemoglobin, myoglobin. All those require some kind of another group along with the protein to work. These are just a few of the type of proteins, like nucleoproteins, lipoproteins, glycoproteins, and you can have glycolipoproteins, a combination of these as well. Phosphoproteins, hemoproteins, metalloproteins, these are working with adjacent metal groups. Very, very complex. Now, there are 4 levels of protein structure: primary, secondary, tertiary, or ternary. The primary one is basically just a long string of amino acids. All it is, a linear sequence. So that's the first level of protein structure. And you just got a series of amino acids with different side chains. And the order of amino acids determines shape. So that primary protein structure is really important because it determines the final shape. Secondary is when we start getting interaction of the side chains. For example, here we can have hydrogen bonding. The hydrogen from the nitrogen and the oxygen from the carbonyl group. And there are 2 types of secondary structures: helix and pleated sheet. And it's just, I think there's a picture here. It could be the helix. It ends up looking like a classic DNA, only it's not DNA, but it's the same shape. Because long molecules tend to form helixes because it's more stable than just a long, skinny strand. These are examples of the helix. Pleated sheets tend to lie side by side. And normally, in a normal protein, there's a mixture of alpha helix and beta pleated sheets together. Something like this. Or much more complex. We can have thousands of amino acids in a protein. They get super, super complex. And because of that, the structure of proteins can be very, very complex. Now, this is an important slide. These are the type of crosslinks that will keep the protein from unwinding. And they vary in strength. So this is a disulfide bond covalent. That's the strongest. So I'm going to put a number 1 by that. Hydrophobic is the weakest. Hydrophobic means that these two are just attracted to one another because like dissolves like. Hydrogen bonding is third strongest. Salt bridge being ionic is second strongest. So what this means, the more strong crosslinks leads to a more stable protein. Excuse me. In terms of heat and pH. So if you have only hydrophobic interactions or crosslinks, you're gonna have a very delicate protein. If you have a whole bunch of disulfide crosslinks, you're gonna have a pretty rugged protein. Like hair, for example, it's pretty rugged. You do all kinds of stuff with your hair and it springs right back. Okay, does everyone understand this? This is real important to get. One, the types of crosslinks, and secondly, the strength. So disulfide being covalently bonded is the strongest. Then we have an ionic bond. Hydrogen bond, and then just a very weak bond between two like structures. In this case, two phenyl groups. This is a repeat of what I was saying before up here. All of this stuff applies to this. So side change determines shape. Solvent determines shape. And because in a body, we're always working with a water environment, you know, no one ever talks about solvent. The solvent is as important as the side change in determining the shape of that protein. Okay, and then the fourth structural unit, we talked about primary, secondary, tertiary. There's also quaternary. That means that if there's multiple units within the protein, those can be positioned and bound in various ways, and the subunits then determine a quaternary structure. That includes, you know, like metal complexes like this. We call them prosthetic groups. I don't like the adjective prosthetic. I think it's an internal part of the protein, so it's not something that's added on as a, I don't know where they got the prosthetic naming of that. Okay. So for example, hemoglobin, it's got 4 subunits, which are proteins. that are, come together. And hydrophobic forces, in other words, like structures tend to go together. And that's what keeps the subunits together. That means it's fragile. Remember, that's the weakest type of bond is the hydrophobic forces. So this unit is very fragile. And then there is a ion prosthetic group in the middle here. So those all form the hemoglobin. Okay, as I've been talking quite a bit, we can hydrolyze protein or denature the protein through pH differences, like we do with ceviche and acid, or we can heat the protein, as in cooking. What happens is the protein is then hydrolyzed to smaller peptides and then eventually to single amino acids. Okay, and in your stomach, that enzyme is called the protease, which amino acids. You've probably heard of pepsin. That's a common protease, but the generic name for enzymes that hydrolyze proteins is called protease. Okay, this denaturation is a big deal with proteins. The protein loses its native state. Are very sensitive. Leads to loss of biological activity. Okay, that means the enzyme stops functioning. Its activity falls off. There's a lot of terms when we get into enzymes about this denaturization process. Now, when you start with a nice protein and it undergoes a heat or pH change, it's going to be denatured, and then it's not necessarily going to go back to that. It can, depends on how much heat and pH change the original protein undergoes. If it's just a tiny bit, then this original structure comes back. If it's a lot, then it's totally changes, unrecognizable. Okay, so typically, these are the conditions that will denature a protein. And these are very genetic. Heat, UV light. That's why UV light is not good for your skin, because it messes up the your proteins in your skin. Change in solvent. Talked about that a fair amount. We all know that acids and bases are not nice on your skin. Detergents mess up the interaction between the solvent and amino acid side chains. And I already mentioned this about heavy metals. Those will bind to the thiols on the cysteine amino acid. And secondly, except for this one. It depends on how much of these you add to the protein. If you do a tiny, tiny bit, your protein can kind of survive, except for heavy metals, because these permanently bind. But these others, you know, if it's a weak exposure to an acid or a base, or it's just a weak solvent, a little bit of light, proteins can usually do okay. But the stronger you get any of these three, or four, I guess, there'll be less and less of a chance of a protein surviving in its native state. All done. Okay, so there aren't a lot of reactions in this chapter. The main one involves amides, both hydrolyzing and forming. That's done the usual way you'd form an amide on the bench in your body. It's done enzymatically. That's basically the main reaction. Okay, so that's what I wanted to cover today, proteins. Now, are there any questions so far? Okay, for those of you who have Wednesday lab, the groups are going to be broken up into, I know when we did the original aspirin lab, the groups tended to be on the larger side. You're going to be breaking up into subgroups because I want groups of 2 or 3 doing the infrared in the instrument room. So between now and when you go to lab at 3, think about your group organization. And it's also going to be turned in together with the first lab. So the synthesis of the aspirin and the analysis of the aspirin are one lab. Okay. All right. And also for those of you who are coming in late on this lecture, I do have your test graded, as you noticed. You'll see your score, and I'll have them back in lab for you. Okay. Are there any questions or anything? You're welcome. All right, so why don't we go ahead and end this?